What's up? I'm Tulio Trevino. This is the Chemistry World. Today we will introduce informative capsule about drugs.
Throughout our lives, we have used hundreds of drugs, either from a simple skin cream or even a pain pill for a headache. We have come to realize how dependent we are on them. However, have we ever asked where the drugs come from? Or what is it that makes them effective in curing some diseases? Well, it all starts with finding a lead compound. Have you ever wondered what makes drugs work, or where do they come from? Drug discovery is a complex, interdisciplinary pursuit of chemistry, pharmacology, and clinical sciences which have benefited humankind immensely over the last 100 years. The drug contains a compound called active principle. A lead compound is a molecule, enzyme, protein, gene, peptide, or antibody that has some kind of pharmacological activity by joining to its target and has an effect on its activator or inhibitor. Regardless of the level of activity or side effects that may occur, it is used as a basis for the development and design of analogs with specific medical properties. It is estimated that in 10,000 compounds synthesized, only one medication is suitable for use. But where can we find a lead compound? Natural products play a dominant role in the discovery of leads for the development of drugs for the treatment of human diseases. Natural products are a great source for the production of compounds that have any type of biological activity. For this reason, most of the leader compounds is obtained from this source. Generally, it is desired to find the active principle, which is part of the lead compound that has a desired pharmacological activity. These lead compounds are usually secondary metabolites. When a plant creates chemical compounds to defend against predators, they are called secondary metabolites. They are called this way because they are not essential to the survival of the plant. The secondary metabolites have a very complex and novel structure which makes it difficult in their synthesis and almost impossible, so it must be directly extracted from a natural source, being a slow, costly, and inefficient process. But based on these structures, we can design a simpler structure. From 1981 to 2010, 1,355 new chemical entities were discovered, of which 28% were of synthetic origin, demonstrating the superiority of natural products. The kingdom of plants. From plants, we obtain the most commonly used compounds. This is why plants are an important source for production of new compounds. One of the first anti-cancer agents developed has been paclitaxel, or Taxol, extracted from the bark of Pacific yew, Taxus brevifolia, and its anti-tumor activity is observed in a broad range of tumors. Oseltamivir is a selected antiviral prodrug against influenza virus. It is produced from the plant of star anise, Illicium anisatum, or from the fermentation of strains of a genetically modified bacteria called Escherichia coli. Unfortunately, there are many species of plants that have not been widely studied, and there are even some that are extinct now. This means that many compounds with biological activity are never going to be discovered, as in the case of Silphium, a plant grown in Cyrene in North Africa, used as a contraceptive in ancient Greece that is now extinct. The microbiological world. Bacteria and fungi produce a wide variety of microbiological agents which are used for drug development. The main drug developed with these microorganisms was penicillin, which led scientists to investigate further the microbial realm. They discovered compounds with pharmacological activity that are unrelated antibacterial therapies, and thanks to these advances, the investigation of microorganisms is becoming more used to searching for compound leaders. A major advantage of using microorganisms is that unlike the plants and marine animals, microorganisms are easy to collect, transport, and cultivate. Lovastatin is used for lowering cholesterol in those with hypercholesterolemia and thus preventing cardiovascular disease. It was isolated from Aspergillus terreus. Romidepsin is an anti-cancer agent undergoing clinical trials as a treatment for cutaneous T-cell lymphoma, peripheral T-cell lymphoma, and a variety of other cancers. Romidepsin is a natural product obtained from the bacteria Chromobacterium violoseum and works by blocking enzymes known as histone deacetylases and inducing apoptosis. The marine world. It was recently discovered that marine organisms such as corals, sponges, and fishes have many biological compounds which exhibit anti-inflammatory, antiviral, and anti-cancer properties. However, the extraction of these marine species must be done carefully to avoid damaging the ecosystem. Cyanobacteria has been one of the richest maritime natural sources of new clinical candidates and lead compounds for the treatment of cancer, and several of these exert their activity through modulation of microfilament polymerization process. Curacin A is obtained from a marine cyanobacterium and shows potent anti-tumor activity. Quercetin A has shown significant in vivo anti-tumor effects in human xenografts, as well as an interesting profile in hollow fiber assays. Significant problems with the instability and poor water solubility of Quercetin A has hindered its development as an anti-cancer lead compound. Because of these ways, scientists have been applying combinatorial synthesis methods to produce more stable and efficient analogs. Another example of marine sources was when it was isolated to two novel pyrimidine nucleosides, espondyridina and sphingosine, that by synthetic modifications led to the first two drugs of marine origin, Ara-C or sitaburina as anti-tumor and Ara-A or acyclovir as antiviral. Cytarabine synthesized from sphingosine. It's a chemotherapy agent used mainly in the treatment of cancers of white blood cells. It's extracted from a sponge. Animal sources. Parts of some animals can have many chemical compounds that can be studied in search of new drugs that generally come from parts of the skin or from their poisons and toxins. These transgenic animals can produce and develop enzymes or proteins. The main difficulty for their isolation lies in that it is in small quantities in the organisms. Epibotania is a potent analgesic and is extracted from the skin of the clawed frog native to Africa. And secondly, transgenic rabbits were developed from which produce interleukin-2 and alpha-glucosidase, drugs used to treat tumors and Pompe disease respectively. Poisons and toxins are widely used as often to interact specifically on macromolecules, allowing to study their receptors, ion channels, and enzymes, obtaining more specialized leading compounds. Clostridium botulinum, neurotoxin, causes botulism and is used for the preparation of Botox, preventing muscle spasm. Animal biotechnology. Genetic engineering techniques allowed obtaining transgenic plants and animals, and also what appeared was the potential use of these organisms for the production of pharmaceutical recombinant proteins. The strategy of using farm animals, sheep, cows, pigs, goats, chickens, rabbits, etc., as recombinant drug products, these factories are called drug farms. Goats produce tissue plasminogen activators and antithrombin 3s, which are employed in the treatment of blood clots that causes diseases such as pulmonary embolisms, myocardial infarctions, and cerebral ischemia. Medical folklore. Formerly, our ancestors performed mixtures of compounds such as leaves, berries, and seeds to verify what effects it caused on sick people. Currently, traditional medicine is not widely used, but these plants or mixtures can serve as a source to find compounds that really have an active principle and from which would serve for the treatment of diseases. Mayan and Aztec cultures of South America use extracts from a variety of shrubs and trees as the roots of Ipecacuanha containing as active principle emetine used as an expectorant, and cinchona bark that containing quinine was used as an anti-malarial agent. Camptothecin was isolated from the Chinese tree Campoteca acuminata. Extracts of these are well known in traditional Chinese medicines for anti-cancer treatments. Isolation and purification. When the lead compound or active ingredient is a mixture of compounds, it has to be isolated and purified. Isolation and purification mainly depends on the nature of the compound. This refers to where it is volatile, soluble, or insoluble, and it also depends on the stability of the compound and the amount of it. When Alexander Fleming discovered penicillin and its antibiotic properties, he succeeded in isolating this in an aqueous solution, but could not purify it because this would be destroyed by the water. Herbal medicine. Herbal medicine contains a large number of compounds that may have different biological activities, and they may bring about side effects or toxics. The effect of the active principle in the herbal medicines is reduced because it is in a lower concentration than in a drug. Polyphenol E is the first anti-cancer that was made from a botanical agent. It was approved by the FDA in 2007 for treatment of genital warts related to human papillomavirus, HPV. It is made with decaffeinated green tea, which contains chemicals called catechins, which are antioxidants, also called green tea extract.
We await you at the next informative capsule of the chemistry world.